So hello everyone, and thank you very much for joining us. I'm excited for the opportunity to present Barcode Diagnostics and the exciting projects that we're currently leading in the field. So Barcode Diagnostics is a company in the field of precision medicine or personalized medicine, and we're currently working on two exciting projects which aim to solve on the long-term cancer a very potential unmet need in cancer. And in the short term, we've been working on COVID-19 and want to share with you the product that we are developing very, very fast in this space to actually help the companies and global markets come out of this pandemic. I'll start by introducing very shortly our cancer-based product and then move on to the COVID technology. In cancer, what we're doing is we're trying to solve a pain that exists for numerous years in cancer, and that is that about 1/3 of cancer patients do not receive the optimal treatment. This is not because the doctors are prescribing the wrong drugs. It's because the doctors don't have currently the right tools to provide the optimal medication for each cancer patient. And for instance, in breast cancer, a physician will prescribe a patient one drug following a CT scan, and this drug will be given for 3 months of treatment following an additional CT scan to actually compare the activity of that drug in that patient. If that drug is active, then the patient will continue receiving it. If it's not effective, the physician will change the drug, and this causes a loss of valuable time which that specific patient doesn't have. And of course, suffering from unnecessary side effects of the less than optimal or wrong medication. And this is exactly the pain that current precision medicine technologies are trying to solve and optimize. And in Barcode Diagnostics, what we're trying to do is actually provide a tool to these physicians, to these oncologists, that can guide them in selecting the optimal treatment. And the way this tool works is actually by sending very, very small doses of numerous drugs to the cancer tissue using nanotechnology. So what we actually do is we encapsulate a drug together with a molecular barcode for that specific drug in a sphere, in a nanoparticle. And in this way, we can generate a mixture of different nanoparticles with very low quantities of drugs. And each drug in each nanoparticle is encoded by a unique identifier or a unique barcode. This mixture is then injected into the bloodstream of the patient. It reaches the tumor, and then once in the tumor, the nanoparticles go into the cancer cells. And if the cargo they are leading is actually an active molecule in that specific cancer cell, then that molecule will signal the cell to die. It will stay, the molecule, the drug and the barcode will be in that cancer cell. If the drug that was in the nanoparticle that entered the cell is not active, then there won't be a signal to die. And what we actually do is we take a biopsy from the cancer tumor, from the tumor tissue. And in that biopsy, there are actually dead cells with barcodes and live cells with barcodes. Dead cells are with the barcodes of the effective medicines, and live cells are with the ineffective medicines. And what we do in our analysis center in the lab is we can actually take that biopsy and we can differentiate the cells. So dead cells go into one vial and live cells go into another vial. And what we do next is we actually quantify the amount of barcodes for each of the specific drugs. So if a drug is active, there'll be more of these barcodes for that specific drug in the dead cell population versus the live cells. And this is exactly how we can generate a potency report to the physician within 72 hours from the injection of these nanoparticles until we can provide the physician and the patient with this report. This is actually how we see the product in the cancer space. So our product will be actually two vials. One, the blue vial is a mixture of the nanoparticles together with the barcodes that will be injected into the patient. And then the biopsy taken 48 hours after this injection would be placed in the yellow vial. And the yellow vial will be sent to our analysis center where we can actually derive the potency of the different medications and provide the physician and the patient with the report regarding this activity of the different drugs so that the physician can treat the patient according to the most active molecules, the most active drugs. And using this method, we can also collect big data sets that can be used not only for the generating the report for each individual, but actually to help drug companies develop additional drugs for cancer. So in terms of the timeline for this type of product, the company was established after a proof of concept was published in Nature Communications, one of the most peer-reviewed papers in the world. The company received seed money from the Israeli Innovation Authority and was established in 2017. Following the establishment of the company, we've met with the FDA to try and understand the regulatory pathway that we need to undergo with this very innovative product. During the last years, we've encapsulated different drugs and biologics in our nanoparticles and produced a GMP production facility, which the company owns. We have raised first round at the end of 2020 and are aiming for a first in human with this type of technology in 2021 in Israel. Now, following the pandemic, the SARS-CoV-2, the COVID-19 pandemic, we asked ourselves, how can we use the know-how and the exceptional team that we have in Barcode Diagnostics and offer our help and our solution to screen SARS-CoV-2. And we've came up with a system that can actually significantly increase the scale of testing by several hundred folds using basically the current tests, but adding a molecular barcode for each of the different individuals. And I'll show you how this is done. By doing this, we can actually reduce the cost per test and reduce the amount of reagents that are currently used in these tests. And this is a known bottleneck currently for COVID-19 testing around the world. The company has already received a grant from the Israeli Innovation Authority to accelerate these the early stages of development, and we're now seeking additional development for this specific product. I'll just want to show you how the current testing is done and how we are providing a next generation testing facility. So currently the RNA is extracted and amplified using one sample in one vial. What we want to do is based on known science. We want to label each and every sample that we receive. In this way, we can generate an amplification process that is barcoded for each individual screening, and we can pool together thousands of tests, one single vials. In this way, we can generate a kit that can be used by the current diagnostic providers to increase the amount of testing that is currently done in these labs from, let's say, hundreds of tests per lab to tens of thousands using the same workforce with some modifications of the system. And this is, we're running very, very fast with this product. We've already talked to the FDA. We're submitting the EUA, the Emergency Use Authorization, to the FDA within the next this week, and have already begun the regulatory and upscaling of this process to be in the market as fast as we can for this pandemic. We have intellectual property in the field, and we have an excellent team that sits in Israel and also worldwide. And this team is composed of clinical scientists, molecular biologists, clinicians, and an excellent board which governs the development of the company. So we are actually in Barcode Diagnostics providing a revolutionary precision medicine products that are changing the outcome of treatment, both in cancer in the long term and in SARS-CoV-2 for viral screening and mass screening and population screening. Thank you very much.
Thank you very much, Ronen, for your presentation. I just wanted to remind everyone that we will be taking questions. We will not be unmuting everyone. So if anyone has any questions, you can either submit them to me in the chat or directly to Ronen, and we'll be happy to answer your questions. On the bottom of your screen, there's a chat function. If you click on the chat function, you can either click on my name or Ronen's name and a private message, and then that'll be the way to ask the questions. I see that we have it. Oh, I got a question over here, Ronen. After FDA approval, what could be a reasonable time to market?
So we're currently working together with the suppliers of our reagents and barcodes. So we're actually working on a system that can enable the kit to come ready for use with barcodes in each different barcodes for individual testing for each of the wells. So this could be actually pooled together for the multiple testing. We're talking to the suppliers that are providing these reagents for the development process in order to keep them on the loop and actually sign agreements even as we develop this before we go to mass screening, so that we can be ready together with them for building up the supply chains. So we believe that once proof of concept has been established and the FDA has been approved, the mass screening kit will be ready for market launch within days to weeks. So the idea here is to have the barcode SARS-CoV-2 diagnostic kit for multiple screening before the autumn of 2020, and this is when the next big the next second big outbreak worldwide is expected, and this is what the people who are in the field are talking about.
Thank you. And we have another question over here. Are other nanotechnology companies exploring the same concept? And how does Barcode see itself primed to be the first to provide this solution?
Okay, so if this is regarding the cancer diagnostic product, this is a unique product that Barcode is developing, and we are, as far as I know, we're the only company in the field that is actually testing the activity of the different drugs directly in the tumor inside the patient. So this provides the right environment to test the drug, the inside the patient's body. Regarding the SARS-CoV-2 product, we know that there are additional companies that, and specifically academic institutions, who have provided a proof of concept for barcoding systems, actually to screen multiple individuals together in a pool. There is currently no approved product in the field for SARS-CoV-2, for the COVID-19 virus, but we know that there are at least 2 companies worldwide who are trying to work on the same technology, and we are currently in talks with the Weizmann Institute in Israel, which is also trying to develop this type of technology. So the notion that additional companies worldwide are trying to work with this type of technology actually shows me that we are on the right track.
Okay, we have 2 more questions here. 1 second. How are the individual samples tagged? Are they one at a time?
Yeah, so the current method is actually to add the tag for the viral testing, to add the tag exactly inside the vial that is extracted from, or in the vial that the swab is placed in. So in this way, the swab can actually pick up the viral construct from the nose. You place it into the testing vial. The barcode and the reagents for the testing are all in the same vial, and in this way, in a short time frame, you can extract the sample that is tagged for that specific individual and place it into a pooling vial. So this could be screened using next generation sequencing. So in this way, you can generate a screen of up to 10,000 individuals using one vial, and you know exactly who has the virus. And it's important to mention that using our technology, we have not only the viral construct that we are screening, but a big question is, can you also include a positive control in each of the different samples? And by using our barcode technology, we can actually validate that each sample that we are taking from each individual is not only tagged, but it also has internal control inside. And that is very important in terms of regulatory approval for SARS-CoV-2 testing.
Okay, we're just going to combine the last 2 questions into one. And it's we have 1 minute for these 2 questions. Who will be the customers for the COVID product? And in addition, another question asked is, which companies are you currently in cooperation with?
Okay, so the customers here are actually the labs who are currently performing the tests. So we can increase the number of tests provided for each of these labs that are doing the COVID-19 testing from let's say an individual lab in Israel that we're working with is testing currently up to 1,000 tests per day, per 24 hours. Using our technology, this specific lab can increase the number of tests from 1,000 to up to 100,000 a day. So we can increase by even 1,000-fold or even more the amount of tests that are tested in a lab that currently uses the same COVID-19 tests. And because our system is based on the current PCR method, there's only going to be a very small adjustment in order to screen the large population. The second question was, I'm sorry?
Which companies are you currently cooperating with?
So we're collaborating with first a lab that is testing in Israel, and we're looking for additional collaborators. We're currently talking to potential collaborators in the States. Basically, also suppliers which can increase the number of kits that we can work with them on constructing in order to go up from initial small development sizes to mass screening of populations. So we can provide thousands of kits and millions of kits for worldwide distribution within a very short time frame.